Hello friends, welcome you all to Bioskul. This is Gayatri. So in today's session, we are going to discuss about GATE 2020 Life Science paper and subject is Microbiology. Yes, it's true that same question, it may not be repeat in your examination, but definitely concept will repeat. If you are a life science student and preparing for any of the competitive examination, it may be the CSIR NET or it may be the ICMR or the ICR, that does not matter. So please do watch the video till the end and get a clear concept. And if you have not subscribed my channel yet, then please do subscribe and stay connected for more such informative videos. So let's proceed. Question number 1. The technique of microbial pure culture was pioneered by. This question, it is very simple and very straightforward question from microbiology. Hope you all know the answer. The answer is Robert Koch. Robert Koch, he postulates that if you compare between a healthy organism and a diseased organism, then in the diseased organism there must be presence of some causative agent or suspected agent. Then that suspected agent or causative agent that must be isolated from the diseased organism and it should be grown in a pure culture media. After that, if we inject or inoculate this pure cultured organism into a suspected healthy host, then that will cause the same disease. Okay. So for this question, the correct answer is Robert Koch. Edward Jenner, he is well known for discovering the vaccine for smallpox. So this one is not correct answer for this question. Louis Pasteur, he is well known for the germ theory. Robert Hooke, he discovered the cell. So the correct answer is Robert Koch. So let's proceed into the second question. The antibacterial trimethoprim, it is an inhibitor of. It is the inhibitor of enzyme dihydrofolate reductase. So trimethoprim, it is basically it inhibits the folic acid biosynthesis pathway. Thereby, it prevents the replication of bacteria. So let's see the pathway first. Dihydropterate diphosphate and p-aminobenzoic acid PABA, sorry, A is missing here, PABM, in presence of enzyme dihydropterate synthetase, it synthesizes the dihydropteric acid. Then this dihydropteric acid, it will convert it into dihydrofolic acid. Dihydrofolic acid converted into tetrahydrofolic acid in presence of enzyme that is known as the dihydrofolate reductase. Okay. So this step, the conversion of dihydrofolic acid to tetrahydrofolic acid, it is inhibited by trimethoprim. Trimethoprim, it is a bacteriostatic. Okay, if it is alone present in the biosynthetic pathway, then it is act as a bacteriostatic. That means it will prevent the replication or the growth of bacteria. But if trimethoprim, it is present in together with the sulfonamides, okay, if it is present in together with the sulfonamides, then that will act as a bactericidal. Bactericidal means it will kill the bacteria. Okay. So for your information, I have here given some of the name of dihydrofolate reductase inhibitor. Here we discussed that trimethoprim, it blocks the bacterial enzyme. Similarly, pyrimethamine, it is a protozoan enzyme, and methotrexate, it is a mammalian enzyme. These are the dihydrofolate reductase inhibitors. Okay, so the correct answer is option 1, that is dihydrofolate reductase. Okay, let's move to the next question. Choose the correct taxonomical hierarchy among the following. So it's the okay, there is a mnemonic for the taxonomical hierarchy, that is did kids pick chocolate over fresh green sprouts? So D, it is for domain, K is for kingdom, P is for phylum, C is for class, O for order, F for family. Here green, the G is for genus, and S is for the species. So let's see. The first it will come domain. Yes, domain, but domain it is present in all the 4 options, and so in the next one, it should be the kingdom, but here there is no kingdom, so it will be phylum, and phylum it is present in all the 4 options. So last it will be the class. So let's eliminate these 2 options. Okay, these 2 options are wrong. And then after class it will be order, but see here it is the family, so this one is also wrong. So the correct answer is 1. The correct answer is option 1. So first it will be the domain, then phylum, then class, then order, then family, then genus, then species. Okay, let's see this one. Shifting a Saccharomyces cerevisiae culture from fermentive to aerobic respiratory mode will. Here it is asking that it is the change from anaerobic mode to the aerobic respiratory mode. Then what will happen? Okay. First we will see, first the glucose in the process of the glycolysis, then it will produce the last product of the glycolysis is 2 molecules of pyruvate. Okay, if the pyruvate it enters into the anaerobic mode of the respiration, then it will first produce the acetaldehyde. Pyruvate it is a 3 carbon compound and acetaldehyde it is a 2 carbon compound. So there is a release of carbon dioxide. Okay, 1 carbon dioxide, as here it is presence of the 2 pyruvate molecules, so there is the release of 2 carbon dioxide molecule. Then in the process of anaerobic respiration it will produce the lactic acid, or in yeast it will produce the ethanol, and during the process of anaerobic respiration only 2 molecules of ATP are produced. Okay, next come to the aerobic respiration. If it will enter into the aerobic respiration, then instead of acetaldehyde it will produce the acetyl coenzyme. Okay, also the acetyl coenzyme it is a 2 carbon compound and there is release of carbon dioxide. Then this acetyl coenzyme it will enter into the Krebs cycle. Then from the Krebs cycle it will enter into the oxidative phosphorylation. So in aerobic respiration it produces the 36 ATP molecule. That means if we compare anaerobic respiration and aerobic respiration, then where more ATPs are produced, it is produced more energy in aerobic respiration. The energy production is higher. Okay, so now see the option. First option is decrease carbon dioxide production. Okay, if there is a more production of energy, then if more production of energy means less utilization of glucose molecule. If there is a less utilization of glucose molecule, then of course there will be decrease in the carbon dioxide production, right? So if it is converted from anaerobic to aerobic condition, then what will happen? There is a decrease in carbon dioxide production. So this option is correct. So let's see the other options. Increase alcohol production. In aerobic respiration there is no production of alcohol, so this one is wrong. Okay, next one, increase glucose consumption. As I told you before that if there is a more production of energy, then why more consumption of glucose consumption it will be decreased, so this one too wrong. Decrease ATP generation per mole of glucose. No, there is high increase of ATP generation per mole of glucose, so this one too is also wrong. So for this question the correct answer is decrease carbon dioxide production. Okay, correct answer is this one and all these options are wrong. So let's see the next question. Which one of the following disease is treated by neuraminidase inhibitor? Okay. So hope you all know that influenza virus it has the two protein, that is hemagglutinin and neuraminidase. Okay. See, this one is the virion or the influenza virus. It has hemagglutinin and neuraminidase. And this HN protein that help in the attachment of virus to the host cell. There is a presence of the receptor. That receptor mainly contains the sialic acid. Okay, so what will happen? This neuraminidase it breaks the sialic acid receptor and that help in the entry of virus to the host cell and that result in the viral replication. Okay, but if there is a presence of the neuraminidase inhibitor, then what will happen? Then it will inhibit the neuraminidase and now the virion it cannot enter into the host cell. Okay, and the viral replication it will stop. So which one of the following disease it will be treated by neuraminidase inhibitor? That is influenza virus. Okay, so correct answer is option 3 and all these 3 options are wrong. So let's proceed into the next one. Okay, which one of following does not provide 3D image? So this question it is from microscopy: atomic force microscopy, confocal microscopy, differential interface contrast microscopy, or DIC microscopy. All these 3 they provide the 3D image. Okay, so all these 3 options it will be wrong, and the correct answer is phase contrast microscopy. Phase contrast microscopy, it does not provide 3D image. It is well known for providing image with high resolution. But if we compare the phase contrast microscopy and the DIC microscopy, that is the differential interface contrast microscopy, then obviously the DIC microscopy, it provides the better resolution. Because in DIC microscopy, the full numerical aperture of microscope is used, whereas in phase contrast microscopy, the annulus of the condenser lens, it restricts the NA aperture, that is the numerical aperture, thereby it the resolution of image, it is reduced. Okay, if we compare the DIC with the phase contrast microscopy, but differential interface contrast microscopy, that provides the 3D image. So for this question, the correct answer is phase contrast microscopy, option 4. Okay, let's see the next one. Which one of the following will increase the resolution of light microscope? So what is the resolution? Resolution is the measure of minimum distance between 2 points of a specimen. Okay, so which one of the following it will increase the resolution? Decreasing the numerical aperture of objective lens. If the numerical aperture it is decreased, what is the numerical aperture? We can say that the numerical aperture, it is the ability of lens to collect the incident light. If there is a decrease in the numerical aperture of the objective lens, then obviously the resolution of microscope, it will be decreased. So this one will be wrong one. Using an objective lens with longer working distance. If the working distance it is longer, then of course the resolution it will decrease. So this one will also wrong. Using a medium of higher refractive index. Yes, this one is correct. If the media has the higher refractive index, it is the measure of bend of a ray of light that pass from one medium to another medium. So if the refractive index it is the higher, then of course the wavelength it will be decreased, and that results in increase in resolution. Okay, so this one is correct. Increasing wavelength of light, no, if wavelength of light it will increase, then resolution it will decrease. So this one is also wrong. The correct answer is option 3. So let's move to the next question. Okay, which one of the following condition favors maximum expression of lac apparent genes in E. coli? So lac apparent means it will utilize the lactose. If in the medium there is a presence of glucose, then of course it will first utilize the glucose as a nutrition, then after it will consume the lactose. So but here it is asking about the lac apparent gene expression. If there is a lac apparent gene expression, then in media they should have the very low concentration of glucose. So here in option 4 there is a glucose high concentration, so this one will be wrong, and this one will be wrong. Okay, so the correct option, either it will be option 1 or option 3. Okay, glucose concentration is low. In option 1 it is showing that lactose is low. If lactose is low, then how can its gene will be expressed more? So this one will wrong. The correct answer is option 3. Glucose concentration low. If glucose concentration is low, then lactose concentration high. If the lactose concentration will be high, then maximum expression of lac apparent gene will take place, and cyclic AMP high. If if glucose concentration will be low, then cyclic AMP it will be high, and it will bind to the cap protein. It will bind to the cap protein. Okay, cyclic AMP it will bind to cap protein, and that enhance the lactose gene transcription. It will enhance the lactose gene transcription. So for the maximum expression of lac apparent gene in E. coli, so the glucose concentration it should be low, and there is the presence of high concentration of lactose and high cyclic AMP. So the correct answer is this one. Is option 3. Okay. Okay. Match the cellular organelle in group 1 with its function in group 2. Let's see. Golgi apparatus. What is this function? Lipid degradation? No. Protein degradation? No. Protein sorting? Yes. The function of Golgi apparatus is protein sorting. Nucleolus? Nucleolus' function is to biogenesis of ribosome. Okay, ribosomal RNA synthesis. Peroxisome? Peroxisome, what is its function? Its function in lipid degradation. Proteasome is the protein degradation. Okay. So the P is 3, P3. Okay, 3 option has P3, so let's eliminate this one. Q4. Okay, Q4 is present in second option. So the correct answer is option 2. And R is 1 is 2. Yeah. So let's see. So this is the last question of part 1 of microbiology paper. In part 2, I'll upload another 10 questions with detailed explanation, and please stay connected for that video too. Okay, let's see this one. A 250 microliter bacteriophage stock containing 8 multiplied by 10 to the power 8 phages per ml, which is added to 500 microliter of E. coli culture containing 4 into 10 to the power 8 cells per milliliter. The multiplicity of infection is, it is asking multiplicity of infection, MOI. Okay, this question is also very simple. Let's see. So what is given? It is given that 250 microliter of bacteriophage stock contains 8 into 10 to the power 8 phages per ml. Okay, so here see the unit is in ml, but here the unit is microliter. So first we have to convert the unit. Okay, similarly here it is the 500 microliter of E culture that contains the 4 into 10 to the power 8 cells per ml. Okay. So what is the formula for MOI? That is the multiplicity of infection. It is the plaque forming unit divided by number of cells. So here it is given that 250 microliter of bacteriophage stock that contains the 8 into 10 to the power 8 phages per ml. So we can write it as 250 microliter, that means 0.25 ml. 1 ml it contains the 8 into 10 to the power 8 phages. Here it is given that 8 into 10 to the power 8 phages, that is present in per ml. Okay, per ml means in 1 ml. Okay, but if in 1 ml there is presence of 8 into 10 to the power 8 phages, then in 0.25 ml how much phages it will be present? Just multiply the 0.25. So it will come 2 multiplied by 10 to the power 8 phages per ml. Okay. Similarly, we will calculate the number of cells in ml. 500 microliter is 0.5 ml. In 1 ml, how many cells are present? 4 into 10 to the power 8 cells are present. So in 0.5 ml, how many cells are present? It is 2 into 10 to the power 8 cells per ml. So now we have to put it in the formula. So the number of phases is 2 into 10 to the power 8 and number of cells is also 2 into 10 to the power 8 cells per milliliter. So what is the answer? It is 1. So the multiplicity of infection is 1. Thank you so much for watching this video. I expect that this video will definitely help you a lot in your examination. If you like this video, then please do share and don't forget to subscribe my channel. Bye bye. See you in my next video.